Welcome to Department of Botany, Kalna College. Myself, Dr. Somnath Mondol. Today's topic in Introduction of Plant Biotechnology, Part 4 is the preparation of golden rice. Golden rice means Oryza sativa genetically modified to biosynthesize beta-carotene in rice endosperm. Now, why beta-carotene? It is the precursor of vitamin A, and the deficiency of vitamin A is called hypovitaminosis A or vitamin A deficiency, which causes severe eye diseases like nyctalopia, xerophthalmia, keratomalacia, and it also defies the deficiency, the immunodeficiency of the patient, leading to multiple infections. A survey by National Nutrition Monitoring Board in the year 2019 showed there are 61% subclinical VAD in India and 49% of whom in Tamil Nadu and 74% in tribal population. This is a survey from WHO long back in the year 1995, which clearly showed the clinical cases of VAD in India. Therefore, in the year 2019, the last survey, we can see there is improvement from clinical to the subclinical cases of VAD. And this is the survey from UNICEF in the year 2018, where we can see in South Asia, including India, 40 to 59% of children aged 6 to 59 months old are received 2 high-dose vitamin A supplements. Now, Ingo Potricus and Peter Baer developed golden rice, and they published their data in the year 2002. Now, this is the pathway of carotene biosynthesis from geranyl pyrophosphate, and in white rice, the very first step of the biosynthetic pathway is blocked due to the absence of phytoin synthase gene, as well as the downstream enzymes are also absent. Now, for making a transgenic rice expressing beta carotene, we need to substitute all these genes into that rice variety. For that, the PSY gene isolated from Narcissus, pseudonarcissus, that is daffodil, that will perform the first step of conversion of geranyl pyrophosphate into phytoin. Next, another breakthrough of the discovery of CRT1 gene from the bacterium Arwinia iridovora, that is an desaturase enzyme, and it will convert phytoin into lycopene. And last, the isolation of LCY gene, the lycopene beta cyclase gene, from Narcissus pseudonarcissus, again from the same daffodil plants. So these 3 genes are important in formation of golden rice. Now, to construct a vector for rice transformation, we need the PSY gene or the phytoin synthase gene from daffodil, CRT1 gene, that is the phytoin desaturase gene from Arwinia iridovora, LCY gene, that is lycopene beta cyclase gene, again from daffodil, the CAM35S promoter, that is the constitutive promoter. We have discussed about the role of CAM35S in our previous video, and GT1 promoter, that is an endosperm specific glutealin promoter. I will come to this in details. And the TP, the transit peptide from P rubisco small subunit, and terminator signal. So they prepared 2 vector constructs In construct 1, within the flanks of left border tDNA and right border tDNA, there are the glutealin promoter, and under glutealin promoter there is PSY gene and the termination signal. Again, under CAM35S promoter there is the transit peptide followed by CRT1 gene and the termination signal. Now, why the glutealin 1 promoter is being used? It is an endosperm specific promoter. Therefore, the gene that is under this promoter will be expressed only in the endosperms. So, in case of golden rice, we need to express the beta carotene, the vitamin A precursor, into the edible part of rice, that is the rice endosperm, not anywhere else in the rice leaves or rice pseudostems. So we need to promote the transcription and expression of all the genes into the rice endosperm. So that is why glutealin promoter has been used. And the transit peptide or TP has been used as it is isolated from the small Rubisco subunits, and small Rubisco subunit that is biosynthesized in the nucleus, and due to the presence of chloroplast localization signal it moves into the chloroplast. Now the whole pathway of lycopene biosynthesis and thereon to beta carotene takes place within the chloroplast. So these enzymes need to transport within the chloroplast. So that is why before CRT1 the transit peptide has been placed. And in the vector construct 2 there are, under glutealin promoter, again the LCY gene, that is the final gene that will convert lycopene into carotene. So these two vector constructs were co-transformed and the Agrobacterium tumefaciens with these two vector constructs were used to infect rice embryo culture for rice transformation. Now, after a successful rice transformation and integration of the tDNA into the rice endosperm cell, and the whole process of tDNA transfer and tDNA integration we have studied in the part 3 of biotechnology study. So suppose here, after a successful transfer of tDNA with our gene of interest into the rice endosperm cell nucleus, now there is PSY gene under the glutealin promoter and CRT1 gene with the transit peptide under CAM35S promoter. And in vector 2, the tDNA region contained the under glutealin promoter the LCY gene. So therefore, after transcription and translation, these three genes will be expressed and they will produce phytoin synthase, phytoin desaturase and lycopene beta cyclase genes within rice endosperm cell. Now, phytoin synthase will convert geranyl pyrophosphate into phytoin. It has an inbuilt transit peptide to enter into the plastid. Next, phytoin desaturase tagged with chloroplast transit peptide will obviously enter into the plastid and convert phytoin into lycopene. And finally, lycopene beta cyclase will convert lycopene into beta carotene within rice endosperm plastid. Thereby, beta carotene will be expressed in the rice endosperm cells, leading to a successful formation of provitamin A, and that will be consumed by us, leading to the makeup of vitamin A deficiency in the VAT population. That's all. That is the picture taken from a Wikipedia. One is the control white rice that we generally intake in our daily diet, and other is the transgenic uh, golden rice where the beta carotene has been expressed. That's all for today.